Let's get started with our meeting today. Welcome. I am John Kelly. I'm an agronomist and I work with Redox Bionutrients. Let's just, I will share my screen and we'll get started our discussion of nitrogen efficiency. Just a brief background. Redox is a bionutrient company. We focus on sustainable plant nutrition. We're headquartered in Burley, Idaho, and we have manufacturing facilities there, Texas, and in Monterrey, Mexico. And we've been in business for 28 years. We focus on four key solution areas: abiotic stress defense, soil health, root development, and nutrient efficiency. In our discussion of nitrogen this morning, let's just get an overview of how plants function. Plants, of course, capture sunlight energy and convert that via the process of photosynthesis to chemical energy. And the chemical form that is a byproduct of this are carbohydrates, which are carbon, hydrogen, and oxygen. Now, this carbon, hydrogen, and oxygen energy source, the plant combines with nitrogen and forms what are called amino acids. And these amino acids chain together to form proteins. And the amino acids are the building block of life. So literally every component of a plant is made of amino acids. While nitrogen is a very, very important nutrient and macronutrient, when we look at the overall plant composition, about 97% of what a plant is made of is just carbon, hydrogen, and oxygen. When it comes to nitrogen, it's a relatively small portion of what a plant's total composition is. However, again, it's a component of each and every plant cell. Nitrogen is assimilated in two basic forms: a negatively charged nitrate form or a positively charged ammoniacal form. And as a rule, it is actually quite easily assimilated by plants. And where does conventional nitrogen, synthetic nitrogen, come from? Early 1900s, just after the turn of the century, two German scientists, Haber and Bosch, they perfected a process called the Haber-Bosch process. And this involves natural gas as a catalyst, heat, pressure, and forms anhydrous ammonia. Unfortunately, the German utilization for this process was primarily to make bombs for World War I and II. But of course, after World War II, this conventional nitrogen went on to promote what we call the Green Revolution, as yields worldwide tremendously improved. It's important to understand, before we talk about nitrogen efficiency and some methodology for improving that efficiency, to understand the whole process. First of all, I'd like to point out that it does not matter what form of nitrogen applied to the soil, whether it's a nitrate form, an ammoniacal form. Eventually, microorganisms are going to convert this nitrogen to nitrate nitrogen. And nitrate has a negative charge. The nitrous monas, nitrobacter, the organisms that convert this, this is fundamental to why nitrogen efficiency is relatively poor. First of all, the soil has a net negative charge, so does nitrate nitrogen. Therefore, there's no chemical attraction. Therefore, nitrate nitrogen is highly susceptible to leaching past the root zone. And of course, that has human health, environmental considerations as nitrate accumulates in groundwater, aside from the fact that we're losing nitrogen that otherwise could benefit our crop. In addition, during the nitrification process, is in these gaseous forms, this nitrogen is highly susceptible to volatilization, particularly if the nitrogen is on the soil surface in the presence of some moisture. That is problematic because for example, nitric oxide has over 300 times the greenhouse gas potency of carbon dioxide. So there are environmental issues, volatilization in the air, nitrogen leaching in the soil, aside from the very important economic effects of not getting nitrogen into the crop where it's going to be able to benefit from a yield standpoint. So most of the nitrogen taken to the plant is the nitrate form, and it accumulates in the vacuole of cells. Now, under moderate conditions or good chemical balance, this nitrogen would go on through a reduction process to an ammoniacal form, and eventually would form amino acids, which chain up and form proteins. Unfortunately, if nitrogen is stored in excess in the cells in the vacuole, it induces chemical changes within the cells and in the plant, produces excess vegetative growth, very weak cells highly susceptible to both biotic and abiotic stress. This is one of the biggest challenges in agriculture, is there are diminishing returns with increased nitrogen usage. So finding that balance and find that nutritional balance is very important. And of course, our objective is that nitrogen becomes incorporated into proteins where it can benefit the plant. I'm highlighting a graphic here that we don't need to spend time on. It's from National Geographic and highlighting the global challenge of nitrogen. And essentially what they're saying is half the nitrogen applied is not taken up by plants. And if half that nitrogen is going into the air or into our groundwater, of course, that has important implications. Well, what can we do about it? Well, I'd like to discuss the first strategy that anyone can accomplish, and it's just a basic practice that is very beneficial. It's what is called developing nitrogen budget. Now, I know it says 8 steps, but just let's walk through these. It's simple and easy to do. First of all, determine what your yield expectation is, and there are a number of excellent apps available that you can download, and you simply punch in your crop, your yield expectation, and it will tell you what the removal quantity of nitrogen is. Next, you determine what you anticipate applying from a nitrogen input standpoint. Now, keep in mind that the more in-season and the more frequent nitrogen applications are going to inherently produce better efficiency applied in more moderate levels exactly when the plant needs it. But step 1, step 2, step 3, if there's any manure applied, determine the amount and the type. You determine how much irrigation you expect to use, and if you know the nitrogen number from that in parts per million, you can determine that. And then it's also beneficial to know your anticipated rainfall during the crop cycle. You put in a leaching factor. The factor, the natural loss can be 50%, whatever you think is reasonable. You can also use a soil test nitrogen, and these are optional. Soil test nitrogen is not a tremendously reliable way to determine nitrogen availability, and you could even include the seed input. The idea of this nitrogen budget is you want to be at or near 100%. If you're at 120%, that would mean that you've got too much nitrogen in the system. If you're at 80%, too lean. So the idea is you want to achieve about 100%. So that's a great place to start. The next strategy is to focus on what we refer to as soil health. This focus on soil health can have tremendous benefits for increasing nitrogen efficiency. And I'm going to discuss three principles of soil health. The first being soil biology, the second will be plant physiology, and the third will be soil chemistry. Let's discuss soil biology. Now, why would improved biology improve nitrogen efficiency? And the answer is very simple. If we have good, beneficial, high levels of microbial activity in our root zone, the microbes, like the plant, require nitrogen. And as this nitrogen, becomes available in a free standpoint, microbes will incorporate this nitrogen into their biological cycles, and it'll stay put in the root zone there. And then we reduce the ability for that to volatilize or to leach. So very, very key component. The second is plant physiology, specifically root growth. Now, naturally, better quantity and quality of roots will have the potential to absorb more of this nitrogen as becomes plant available. But in addition, 90% of the beneficial biology in the soil occurs within a couple centimeters of the root surface. Therefore, better root system, better quantity and quality of root system inherently is a better platform where you become more efficient with nitrogen. The third component of soil health is soil chemistry. Now, soil chemistry, from many different standpoints, is important, but one of the key components of soil chemistry is its impact on soil structure. And when we refer to structure, we're referring to the relationship of macro and micropore space, where in ideal air-water relationships occur. Not only does that impact biology in a beneficial manner, impact the potential for roots to grow, it also helps deliver nitrogen to the root zones more efficiently. So again, this concept of improving soil health can have a profound impact on nitrogen efficiency. With that in mind, I'd like to introduce just a few product technologies to give you an idea how we can implement these practices. The first product I'm going to discuss is called Root RX. Now, Root RX is a botanical extract, and what it does is it stimulates root growth and plant root metabolism, particularly under very adverse conditions for root growth. This is a product, when incorporated into a comprehensive, well thought out nutritional program, can really have dramatic benefits on crop performance. The second product I'd like to talk about is H85. Now, H85 is designed to improve soil microbial activity, and not only improve microbial activity, but a broad range of microbial activity. So it incorporates short, medium, and long chain soluble carbon. So we get great diversity under a broad spectrum of soil chemical conditions. And as I'll show you in a moment here, this can have important implications. A third product I'd like to talk about is called Dermaplex Plus. Now, this product incorporates the benefits of microbial stimulation, including the short, medium, long chain soluble carbon, but also contains key metabolites from a fermentation process of trichoderma. We use one specific trichoderma species, and these metabolites stimulate root growth, metabolism, soil health. It's a very quick response in the soil. So this is very beneficial. Now, how would we implement these, and what could we expect to see? What would the benefit be? Well, let's start with Dermaplex Plus. If our objective is a 30% nitrogen reduction, first and foremost, we need to be sure that when we're reducing from what? If we're already very efficient, very lean, we cannot expect that high of a response. If we're over applying nitrogen, our anticipation would be it could be even more efficient. But at 100% nitrogen budget, where you've done the budget, you know where you're at, you can assume a 30% reduction in N requirements, utilizing this product at 0.15 gallons for every 60 units of nitrogen. And the best results are going to be application of this with your nitrogen or at the same time as you're applying your nitrogen. The second product, H85, again, you can assume a 30% reduction, again, at that 100% nitrogen budget with a quart for every 60 units of N. And the third is Redox. You can look for a 30% reduction at that 100% nitrogen budget, where a half gallon per 100 units. Now, you might ask, well, which of these to use? Well, first, speaking to a Redox agronomist will help you determine, but I will say the very most common is to use a little of all three. That's the most common approach. Another technology I'd like to discuss goes back to the nitrate nitrogen storage in the vacuole when that becomes excessive. During that key stage of plant metabolism where that nitrogen needs to be reduced to an ammoniacal form to form amino acid and proteins, we have technology called Supreme. And Supreme helps improve this nitrogen metabolism. So we apply this during these reproductive stages of growth when we wanted to set and retain more crop. So very, very effective tool for that. All right, in just the couple minutes that I have remaining, I'd like to discuss I'll just go over two quick results. The first one is on celery, was done in South Central coast of California by a third-party researcher using RootRx. And let's get to the chase. If you look at the first column on the right, that is utilizing CAN 17, assuming 100% budget and just straight CAN 17. As you can see, we get our yield, very good yield. The second column is 100% budget using RootRx. Again, is the product for improved root growth and metabolism. And we get a modest 8% increase in production. The most interesting thing is in the third column is when we reduce that nitrogen budget to 50%, what happens with straight CAN 17? We see a sharp reduction in productivity. However, when we have applied a 1% solution of RootRx with that CAN 17 in the fourth column, what do we see? We see no yield loss from 100% and actually a very small increase. It just highlights the potential of the role of that root system in improving nitrogen efficiency. The second one I'll refer to is Dr. Drew Hubbard. This was done in Oregon. It's a nitrate leaching study. And very quickly, what they found is when you take straight nitrogen in the form of urea, you can see that follow that yellow bar. What that is showing is the amount of nitrate that is leaching past the root zone. And you see a lot of leaching, a lot of losses occur. The blue line is a compilation of a number of what we call slow release or nitrification inhibitor technology. What that does, it inhibits the ability of the ammoniacal nitrogen to convert to nitrate. And of course, you can see a more gradual release. The key thing I'd like to point out is when we took H85 with urea, it parallels very much that slow release pattern due to the microbial activity. The benefit of that, of course, is you look to the right, straight urea versus urea and nitrogen. There's a very nice economic advantage, good ROI from that. I hope that this presentation has been helpful. And if you have any comments or questions, I can take those at this time.